Biomedical scientists and engineers interested in implantable devices learn from advances in cell biology, molecular biochemistry, etc., and transfer that knowledge to applications pertinent to implantable materials. Lectures 15A and B and Lecture 16A presented some of those developments and information. The rationale for such research endeavors was the need to make the synthetic biomaterial, which is part of the implantable device, as close as possible to the native natural extracellular matrix. That particular environment is needed in order to promote the interactions of cells from the surrounding tissue at the implant injured site, and it is absolutely necessary to assure success of this particular implantation. The field of chemical modification of the surfaces of implantable materials in order to imitate the native extracellular matrix started with applications of current knowledge regarding how proteins adsorbed on the surfaces of biomaterials and how subsequently cells interact with the pre-adsorbed proteins. Since adhesion to substrates is crucial for the survival of anchorage-dependent cells, this particular cell function was the first one to be addressed in pertinent research endeavors. I will present some examples of pertinent applications from research which was conducted in my laboratories and accomplished by my former graduate students. My research team has conducted experimental studies. They are also known as in vitro studies, using cultured mammalian cells, exclusively in vitro models, and of course examples and samples of various materials and biomaterials. The knowledge that proteins mediate the adhesion and subsequent function of cells was used as follows. Proteins which are known to mediate the adhesion and functions of cells from a specific tissue were chosen and used for the chemical modification of the surface of a synthetic material. The first example is that of a neuroprosthesis, also known as electrodes. These particular prostheses are inserted in the brain for one of two purposes, namely to either record the electrical signals inside the brain or to be used to provide electrical stimulation in specific regions of the brain, either for the purposes of research or for the purposes in the clinics to stimulate the part of the brain of a patient who is suffering from a particular disorder, hoping to improve the function of the brain and improve the health condition of the patient. The brain contains many cells. Among them are the neurons. These are the cells which transmit electric signals and also respond to electrical stimulation from the outside world. Many other cells in the brain are the astroglial cells. In contrast to what the neurons do, the astroglial cells do not transmit electrical signals and do not respond to electrical stimulation. A schematic representation of an electrode is shown the photograph on the right-hand side of this slide. Please keep in mind that this representation is not to scale. In fact, electrodes which are used for pertinent research in the brain are in the order of a few millimeters in length and perhaps 1 or so millimeter width. In addition, I would like to call your attention to the fact that the surface of the electrode is not uniformly covered with the electric circuit. The electric circuit, as schematically shown here, has pads and it has these lines which of course connect the particular system and are used for the transmission of the electrical signals. Another interesting and important aspect is the fact that the edge of this electrode, the part that is inserted into the soft tissue of the brain, is a pointed arrangement. The histology photograph, which is shown on the left-hand side of this slide, shows the results of an experiment after the electrode was removed. You see, stained in red, the brain tissue which predominates in this photograph. In the middle, you see a dark triangular region. Keep in mind that this particular photograph has been produced in great magnification. The triangular area is exactly where that pointed edge of the electrode was inserted, and of course, at the end of the experiment, it was removed. The stain of the brain tissue is pretty much uniform across the field, but if you come close to the insertion site, you see not only a difference in the hue of the color, of the red color, but in addition you see regions which appear to be white or yellow. A subsequent analysis at the cellular level of this particular region reveal that these white-yellow areas are the areas which contain exclusively astroglial cells. As a result of that, and if now the astroglial cells cover the inserted electrode, the outcome is a failure of the experiment, namely no transmission of electric signals from or to the brain. The results presented on the previous slide were obtained by a group of researchers who were working on a pertinent project using animals, experimental animals, at another academic institution. This and related data became the basis for a collaborative interaction. My research team provided the in vitro experiments pertinent to biomaterials which are used or could be used for electrodes implanted in the brain. This particular project was chosen by one of my graduate students, Lance Kam, who completed successfully his doctoral training, and I'm very proud of him. He is now full professor of biomedical engineering at Columbia University in New York City. Lance chose to investigate the chemical modification of the surface of materials using proteins. Another challenge that Lance chose to incorporate into his project was the use of micro-pattern surfaces. At that time, this was a frontier in the biomedical, biomaterials related area. The micro-patterns are formed as follows. You need at the beginning to have a topological master which has the negative impression of the pattern that you are after. You bring an elastomer stem, which is a plastic, and you attach it on the surface of the topological master to form the impressions which you need to transfer. The elastomer stem, the area down now that has the imprinted pattern, is painted with anything you wish. In the case of Lance's experiment, the appropriate proteins. It is placed very carefully on the surface of the material which you wish to pattern, placed there and not moved, not shaked, not wiggled for a period of time, and then very carefully removed. What one obtains at the end is the so-called pattern silicon on this particular slide. And what you can see now, you have a surface says of a certain pattern which are of the original material, but surrounded those patterns you have some other regions which can be modified chemically. Lance chose to use hexagonal patterns for the purpose of covering uniformly the entire surface area of his sample biomaterial. I will not go into the details of the various parameters of the patterns which Lance investigated. Some of the features are listed on the small table under the big figure. If you are interested, you can check the publication which resulted from Lance's doctoral thesis research for further details. The important thing that is pertinent to the chemical modification of the surface of the substrate now is the following. Two proteins were chosen to modify that surface. Both proteins have huge and difficult to pronounce names, so allow me please to use the abbreviated letters DETA for one of them and OTS for the other. What is the important difference? DETA was used to be absorbed in the center of these patterns. DETA is a chemical compound which promotes the adhesive interaction of cells. In contrast, OTS, which was used to paint the perimeter of the patterns, is a protein which does not promote adhesion of cells. As a result of that judicious choice, we have on one and the same surface area, chemically modified now, two regions, one in which the cells may adhere and another one which does not allow adhesion of the cells. The experimental conditions used in this study are listed on this slide. I mentioned the surface modification with OTS and DETA. The cells we first used separately were the astroglial cells. The culture conditions were traditional culture conditions with the appropriate medium for the cells supplemented with serum. The experiments were conducted under standard cell culture conditions, which are 37 degrees temperature, humidified environment, and also an environment that contains 5% CO2. This is a condition that culture cells like, and they are used throughout the cell culture experiments. The experiments using the astroglial cells lasted 6 hours, and they were, of course, adhesion experiments. Subsequently, the cells were stained appropriately and visualized using confocal microscopy. The visualization of the modified substrates with the adhering cells was done in such a way that you can see the difference between the regions which were modified with DTA and the cells adhere, and the regions, the perimeter of those patterns where the cells did not adhere. As I mentioned earlier, Lanz explored various parameters pertinent to the micro-patterned motifs on the modified surface. I'm showing here the results of an experiment in which squares were the patterns used. The interior of each square was the one that had DTA, therefore it was a region which the cells recognized as adhesive, while the perimeters, the outlines of each square, were not adhesive. They were modified chemically with OTS, the protein which does not promote cell adhesion. The important result illustrated on this slide is the following. By playing with the relative surface area which one provides for the cell adhesion, one can literally direct one single cell adhering in one of these squares on the micro-patterned surface. This is a very important result that shows that with the appropriate chemistry, we biomedical engineers can control where the cells will be directed to go and to adhere. This slide shows representative results regarding cell adhesion and morphology when astroglial cells are interacting with the hexagonal micropattern surfaces. The parameter of interest in this study is the dimension of the center of the hexagonal patterns, which was of course chemically modified with the adhesive protein. Details regarding the dimensional parameters of the micropattern motifs are shown at the bottom of each one of the three panels. The important thing to keep in mind again is that only the center, which is shown a little bit darker on this micrograph, the center of the patterns is where the cells may adhere. When that particular adhesion region is very, very small, you notice that the cell cannot be accommodated inside that particular region. So how does the cell accommodate itself? By extending finger-like projections, and as you notice, if you take any of these examples, the end of those finger-like projection is attaching in the center of the micropattern surface, which has the adhesive protein. If we go from the panel, which I just described, on the left-hand side to the one on the right-hand side, when now the interior region of the hexagonal pattern is much, much bigger, it can accommodate single cells, and you can see them filling selectively and exclusively, more and more so, the center of the micropattern surface. Definitely, if that particular surface area is increased, many more cells would interact and adhere in these particular regions, and perhaps many more cells will adhere within one and the same region. In parallel and separate experiments, Lanz investigated the adhesion and function of neurons on the micropattern surfaces which he had established for his project. As a reminder, I'm showing again the schematic of the neural prosthesis node to scale. The challenge here is a little bit more demanding because if we want the neurons to interact with the circuit, we have to find appropriate patterns and modification of the surface of the material, which will allow the neurons not only to adhere, but also to function. In terms of signal transmission in neurons, we know that the body of the neurons, the so-called soma, extends finger-like projections, the dendritic arrangement, out of which one of them becomes an extended arm-like projection with which one neuron contacts the other, and they form that linear pattern which now provides the venue for the transmission of the electric signal. This is a challenge which had to be met with a pertinent chemical modification of the substrates interested in the project that Lange was conducting. In order to promote the adhesion of neurons, which are totally different cells than astroglial cells, we had to learn, obviously from the literature, what are the proteins known to mediate the adhesive interaction of neurons. And in this particular case, it turns out that the protein they like is laminin. And it's not only laminin, it has to be conjugated. It has to form a complex with polylysine. That, therefore, was the protein which was used to modify the regions where we wanted the neurons to adhere. The cells used for these experiments were hippocampal rat neurons. The cell culture conditions were use of an appropriate medium, and the time of the experiment, the duration of the experiment was 1 day. Visualization at the end of the experiment was obtained using scanning confocal microscopy, which had visualized the specimens in 2 regions, differently stained. One was the area where the cells adhere, and the other was the area on the micropattern surface where the cells did not adhere. The process used to prepare the micropattern substrate surfaces, which were later on used for experiments with neurons, is illustrated schematically on this slide. On the left-hand side, we have a reminder that laminin had to be first conjugated polylysine, and that particular complex was then used to paint or stain the side of the elastomer stem which had the pattern of interest. The elastomer stem was then placed carefully and maintained without disturbance on the surface which we desire to micropattern, and at the end a pattern was obtained as shown on the lower hand side of this particular slide. I wish to remind you of a major difference between this now micropattern surface and the one that I described regarding the experiments with the astroglial cells. In the case of the astroglial cells, the interest was to modify the certain of the hexagonal patterns with an adhesive protein which the astroglial cells recognized and adhered on that part of the pattern. In contrast, in the experiments with neurons, the micropattern surfaces had their adhesive polylysine conjugated laminin on the perimeter of the hexagonal patterns. The interior surface of the hexagon did not contain any protein which the neurons recognized and eventually interacted with. This slide shows representative results obtained when neurons interacted with chemically modified surfaces using, of course, the laminin polylysine conjugate on hexagonal micropattern surfaces. A reminder again, the laminin polylysine conjugate, which the neurons like, recognize, and attach, is provided on the perimeter of these hexagonal patterns. The three panels illustrate results obtained on paths which had different widths. Starting on the left-hand side, the path width was 7.5 micrometers, quite wide, in fact. What we see on this particular panel in green is a neuron. You see what we call the soma, or the body of the neurons, and a few extensions like fingers, and those are typical of the dendritic extension of a neuron. One of those extensions, however, is the one that becomes the axon, and this was the function of the neurons which was investigated in this study. As you can see, the axonal outgrowth is on the path and extends to a long distance from the soma of the neuron. Directed axonal outgrowth was also observed when the neurons interacted on a micropattern surface whose path width was 6.7 micrometers. You can see again the green body of the neuron and the axonal outgrowth. The most impressive results, in my opinion, are illustrated on the right-hand side panel. The path width in this case was the narrowest one investigated in the study. Specifically, the width was 2.6 micrometers. What is fascinating for me is the fact that the axonal outgrowth follows very faithfully the outline on the perimeter of the patterns, which contain, of course, the adhesive conjugate, and follows it all the way, and it extends beautifully in a specific direction and at a great distance from the soma of the adhering neuron. These results illustrated that by choosing judiciously the proteins which mediate the adhesive and functional interaction of cells from specific tissues, we can incorporate those proteins on the surface of materials, maintain their bioactivity, and show that this particular chemical modification affects select functions of the cells, starting with adhesion, which is of course of crucial importance in the case of anchorage-dependent cells because their survival depends on their adhesion. But in addition, this particular chemical modification can bring about functions which are pertinent to a specific tissue and the predominant cells of this tissue, in this case, the neurons of the brain. In the meantime, advances in the biological sciences, such as cell biology, molecular biology, biochemistry, etc., provided evidence of the mechanisms which are involved when mammalian cells interact with proteins. Some of these aspects were discussed in a previous lecture, Lecture 15b, with the title Protein Interactions with Substrates. The important lesson from these advances was that cells do not interact with the bulky, big, convoluted molecules of proteins. They are specific domains, or so-called epitopes, composed of a few amino acids those particular groups are called peptides. Therefore, peptides which are the so-called adhesive peptides, and the cells with the receptors on their cell membrane recognize those epitopes and interact specifically with the adhesive peptides on various proteins. These lessons from biology prompted my research group to investigate the following. Chemically modify the surfaces of materials using adhesive peptides, not the big bulky protein molecules. And then, once the surface of the material was modified, to study the adhesion of mammalian cells with those surfaces. For the purpose of research pertinent to the interests of the present lecture, peptides were used in one of the following ways. Immobilized peptides such as RGD. Chemical procedures were used for this purpose, not only to immobilize the peptide on the surface of a specific material, but also to maintain the bioactivity of that peptide in order to assure the subsequent adhesion of cells on that substrate. Another way that peptides were used is the following. Peptides such as RGD were put in a solution, usually a buffer, which contained a suspension of cells. The RGD peptide adhered on the membranes on the cells, the receptors on the cell membranes, and therefore blocked that receptor. For example, the integrin receptors on the cell membranes. Once those receptors were blocked, subsequent attempts to promote adhesion of those cells on any substrate was disrupted, prevented, completely blocked. A partial list of the chemical procedures which have been used to immobilize peptides on material surfaces is given on this slide and includes covalent tethering, photolithography, microfluidic patterning, all the way to three-dimensional printing. The chemical technique which was used to immobilize peptides on the surfaces of materials during the research projects conducted in my laboratory is schematically illustrated on this slide. This chemical technique took advantage of the presence of the OH group, the hydroxy group, on the surface of materials, and subsequently brought in a process which is called silination. In this particular case, this particular group presented free an amine group, the NH group here. When the peptide, let's say RGD, was presented to this now modified, chemically modified surface, the amine group of the silinated surface interacted with the carboxyl COOH group and resulted in this particular covalent interaction of the peptide on the chemically modified surface. The peptide immobilization technique, which I just described briefly, was used by Meredith Hassenbein, another graduate student in my research team, who used this particular technique to study the adhesive interaction of osteoblasts, the bone-forming cells, for her master's degree thesis. This project required knowledge of the mechanism of osteoblasts interacting with the respective proteins which are part of their extracellular environment. In the case of Meredith's project, this information came from research accomplished by other students in the laboratory who had already completed their projects. Important details of the mechanisms underlying osteoblast adhesion on substrates are shown on this table. First and foremost, the cells have membrane receptors. Some of them are the integrins, which recognize on various extracellular matrix proteins such as collagen, vitronectin, etc., the sequence arginine glycine aspartic acid, RGD. This particular mechanism was elucidated by Dave Puleo, another student who completed his doctoral thesis in my laboratory. This mechanism, which involves integrin receptors recognizing the RGD domains, accounted for approximately 45 to 50% of the adhesive mechanism of osteoblasts. There was another mechanism which accounted for approximately another 45-50% of the underlying mechanism. And this mechanism involved receptors which are called heparan sulfate proteoglycans, which recognize different domains on the extracellular matrices. And these domains are called heparin binding domains. One sequence which describes this particular amino acid sequence is lysine arginine serine arginine, abbreviated KRSR. For this reason, immobilization of peptides on a particular substrate had to be investigated from the perspective of the immobilized RGD and KRSR in order to determine which of the two mechanisms is either the predominant and or the one that makes the difference in the adhesion of osteoblasts on extracellular matrix components and, of course, on the chemically modified material substrates. For her master's thesis, Meredith Hassenbein used micropattern surfaces, which were prepared in the similar manner which I described when I gave you the example of the neural prosthesis. Please keep in mind that the patterns now were different for this particular study. Specifically, circles were used, and in the insert cartoon on the lower right-hand side, you see the circles with diameters which were different for the various studies, from 10, 50, 100, or 200 micrometers. In addition, the distance between those circles, indicated by the letter L, always stayed the same at 10 micrometers. In terms of the chemical modification, this interior part of the circles was modified, first with DTA and then with the immobilized peptide, either IgD or KRSR. The outside part of the circles was chemically modified with OTS. Remember from the example regarding neural prosthesis, this is a chemical compound that does not promote adhesion of cells. The experimental conditions for Meredith's experiments are summarized on this slide. First and foremost, neonatal red calvaria osteoblasts, the bone-forming cells, were used, and one more, so to speak, additional detail for this study was the use for parallel but different experiments, the use of rat skin fibroblasts as a comparison line for the functions of osteoblasts versus those of fibroblasts. The culture conditions were appropriate medium, standard cell culture conditions, again a reminder that involves 37 degrees temperature, 37 degrees centigrade temperature, a humidified environment, 5% CO2. The respective cells were allowed to adhere on the substrates for 4 hours, and then the results were visualized using both and or fluorescent microscopy and some cases and light microscopy to visualize the patterns and the cells on the modified substrates at one and the same time. Some interesting results obtained from these studies are shown on the 4 frames of this slide. Looking at the top slide on the left column, we notice that when the diameters of the patterns, the circles which were modified with the adhesive peptide, were 10 micrometers, the smallest dimension checked in the study, very few cells, there is one down here almost on the extreme bottom left-hand side, only a few cells adhered on the modified substrate. This study proved that we need to have a particular size of the total area which is modified for the cells to adhere. You notice there are many more cells adhering on the micropatterned and chemically modified circles which have a diameter of 50 micrometers. Many more cells, and they form beautiful colonies, were seen on the patterns which had diameters of 100 micrometers. And definitely we see beautiful colonies of the osteoblasts on the circles modified with the adhesive peptides and proteins, which had circles which had a diameter of 200 micrometers. Because the visualization procedure could accommodate only one colony when the circles of diameter 200 micrometers was used, we chose to illustrate the results of this study with the patterns which had internal diameter of 100 micrometers. As you can see, in this case, the lower frame of the left column, you can see 4 such circles on one and the same frame of the illustration. A series of very important results were obtained from this study. I will start with the results obtained when osteoblasts were used to interact with the surfaces modified with RGDS. On the frame under fluorescent micrographs, you see the circles of the micropattern. On the photograph, or I should say the micrograph next to it, the light micrograph, you see on the same frame the osteoblasts which are adhering inside the circles which were chemically modified, and they are forming very nice colonies. In contrast, if you use fibroblasts for a similar experiment, remember, only the interior part of the circles has been modified for the purposes of cell adhesion. You can see on the light micrograph with the fibroblasts that cells have adhered on the interior part of the circles. The colony formation, of course, looks totally different. The important conclusion from these experiments is that 2 types of cells, which we consider competing cells in the case of new bone forming around bone implants. We want osteoblasts to interact with the implant and form new bone in order to stabilize the implant and have a desirable outcome. Fibroblasts, when they interact with implants, they form a relatively soft tissue. That is not what is desirable in the case of bone implants. Bone implants require the formation of new bone tissue. In addition to osteoblasts and fibroblasts, as I mentioned a few times in the past, many cells have the appropriate receptors to recognize the RGD peptide, which is part of many adhesive proteins of the extracellular matrix. If we want now to modify the substrate surface of a material which is going to be used for an orthopedic dental implant, the design objective should be to promote the osteoblast interaction and not the fibroblast. The project completed by Meredith proved that we have such an option which we can use and exercise. And that now will be the case of using the other peptide, KRSR, to modify the interior of the micro-patterned circles. Again, on this column, you see the background of the patterns, and from the same frame under light micrographs, you see the osteoblasts adhering inside the circles and forming very, very nice colonies. In contrast, when the fibroblasts are placed on these patterned surfaces, which have been modified, the interior of the circles with KRSR, what we have as a result? Anecdotal, random, and very limited interaction of fibroblasts with those modified surfaces. This is a result which gives us a very important information. We need to judiciously choose the peptides which promote the interaction of a specific type of cells which is needed for the formation of the new tissue which is desirable. Namely, in the case of orthopedic dental implants, bone implants, we want the interaction of osteoblasts and not of fibroblasts. Immobilization of adhesive peptides on the surfaces of implant biomaterials attracted the attention of many biomedical engineers and resulted in many studies in laboratories across this country and across the world. Some of the results of these particular studies will be shown in the following slides. The parameters which prove to regulate the cell responses to immobilized peptides on material surfaces turn out to be some of the following. The type of the peptide, how much of the peptide is used, which we call concentration or density, spacers. Remember, the peptides are composed of a few amino acids. As I said before, 3, 5, 7 maximum. They are very, very small entities, and sometimes when immobilized on the surface, they are too close to the surface and not far enough above the surface for the subsequent interaction with the receptors on the cell membrane. For that purpose, what researchers in the field did is to use what we call extensions, and that meant that we added more amino acids which did not have adhesive properties, but at the very end, at the top, the one area which comes in contact with the receptors on the cell membrane, that was the place where the RGD or the other respective peptide was immobilized. How the peptides were distributed spatially on the surface of the modified substrate has proven another important parameter, and it needs to be addressed for successful outcome of these procedures. Last but not least, I mentioned that there have been many immobilization techniques which were developed and used. The results of certain immobilization techniques proved to be more successful as far as the outcome is concerned, more successful than others. The research which produced chemically modified material surfaces in order to promote specific functions from cells was successful because the immobilized bioactive compounds promoted and controlled cell adhesion and motility and other functions. In such cases, the cell function was specific and was regulated locally. The techniques used for chemically modifying material surfaces, however, had certain limitations as well. One important limitation was the fact that the chemical processes did not give the experimenters control over the presentation of the peptide on the surface, how the peptide was distributed and at what concentration or density, and most importantly, sometimes they do not provide a stable bioactive peptide for the subsequent interaction with cells. This lack of specificity, because many receptors are expressed on many cell types, some of them the same, some of them different. I gave you an example of the similar receptors on cell membranes, on osteoblasts and fibroblasts, which recognize, let's say, the RGD domain. That lack of specificity contributed to the difficulties in the applications of the pertinent chemically modified material surfaces techniques. In the similar perspective, we know now that the big protein molecules have more than one domains or epitopes of peptides which are bioactive, and they bring about functions of cells and sometimes of more than one type of cell. That causes lack of specificity for the outcomes which are needed for new tissue formation. Since cells are living components, they produce several chemical compounds. Some of them may have the ability to break down chemical compounds. Keep in mind that proteins and peptides are very sensitive chemical compounds, sensitive to conditions of their environment, such as pH, temperature, etc. For this reason, some of the chemical compounds which are produced by cells may cause denaturation, breaking down of the proteins and the peptides which researchers have immobilized on material surfaces. Last but not least, this type of research produced substrates which were used in several animal models with limited success. This kind of research was not tested in human subjects. The importance of adhesion for anchorage-dependent cells, their adhesion either to extracellular matrix proteins or to implantable materials, cannot be overemphasized. This function is crucial for the survival of anchorage-dependent cells. Once, however, these cells survive, they need also to function, and other functions include those which bring about the formation of new tissue formation. Formation of new tissue is very important, I will say equally as critical, in the process of the successful outcome of implant biomaterials and devices. This slide, which we have seen several times so far in the course, lists the cell functions which are pertinent to wound healing and tissue regeneration. We started with an in-depth discussion of the adhesion of cells to the substrate. You can see all kinds of other functions which are important, but I would like to bring to your attention the results of a study from my laboratories which address the aspect of cell proliferation. Cell proliferation is needed in the process of wound healing and is a very important aspect in the so-called proliferation repair stage of the tissue wound healing process. The few cells from the surrounding tissues at the injury site are not enough to bring about the restoration of the tissue and the formation of the new tissue. Therefore, many more cells are needed, and that outcome is achieved by the process of cell proliferation. This project was motivated by the need to cover a particular material surface with normal, healthy, intact endothelium in record time. Of course, such an outcome is needed in cardiovascular material and implant devices, for example, the vascular grafts. This particular study used, of course, culture endothelial cells and modified surfaces. For the chemical modification of the material surface, we use the information that the endothelial cells have integrin receptors which recognize a particular domain of laminin, a protein of the extracellular matrix of the vascular tissue. The sequence, the peptide sequence in laminin, which the integrin receptors of endothelial cells recognize, is tyrosine, isoleucine, glycine, serine, arginine. The one-letter abbreviation of this sequence is YIGSR. It is a 5-amino acid peptide. This peptide was used and it was immobilized on a substrate on which subsequent endothelial cells were presented and their adhesion was studied. Immobilization of the peptide from laminin followed the process which I described earlier for the immobilization of the RGD peptide and the subsequent use of those modified surfaces in the study of osteoblast interactions with those modified substrates. The important thing is that the outcome now has a covalently bound peptide on the surface of a material. Pertinent details for the endothelialization of substrate experiments are given on this slide. The cells used were bovine pulmonary artery endothelial cells. The substrate used was glass. I would like to provide an explanation. This study was a proof of concept one. Therefore, glass was used because it is an inexpensive disposable substrate. Because of its transparency, it allows visualization of cells attached on the surface using light microscopy. It doesn't require a more fancy technique like fluorescent microscopy or confocal microscopy. Last but not least, glass has on its surface the OH groups, which, if you recall the slide for the chemical technique used to immobilize peptides, those are the starting point for the subsequent silination and immobilization of the adhesive peptide. The peptide chosen for this study was the one from laminin, which endothelial cells recognize tyrosine, isoleucine, glycine, serine, arginine. Modification of the surface of the substrate via chemical procedure to make it more attractive to the adhering cells was not the purpose of this particular study. What we wanted to promote was proliferation. And as you may recall from other lectures, cell proliferation is stimulated by growth factors. Various growth factors were investigated for this study. The results that I will show were obtained with basic fibrillous growth factor, were the optimal, at a concentration of 1 × 10⁻⁹ grams per ml. The experimental condition was cell adhesion and proliferation under standard cell culture conditions. The experiments lasted for 72 hours. Visualization of the results was obtained using light microscopy. The cells, remember, all the cells, with the exception of the red blood cells, have no color. The endothelial cells had to be stained, and the stain used is crystal violet. Several important results were obtained from this study, and they are illustrated on 4 micrographs presented on this slide. We start with the first frame, which is cell adhesion, endothelial cell adhesion on unmodified glass in the absence of any growth factor. You can see the cells stained with the crystal violet stain. You can see several cells which have adhered on the surface of the substrate. What you should keep in mind, however, is the following. A lot of the substrate surface is bare. It doesn't have any cells covering it. If, and since I should say, the desirable outcome of the endothelialization experiment is to cover completely the surface, we need healthy, intact, completely covering endothelium. This particular outcome is not desirable. Since a modification, appropriate modification, chemical modification of the substrate with the well-chosen adhesive peptide is known to improve the adhesive interaction of cells, the next step in the study was to modify the surface of the substrate using the adhesive peptide from laminin, which is recognized by endothelial cells. Again, these experiments were conducted in the absence of a soluble growth factor. Comparing the 2 frames, left and right, we see that when we have modified the substrate surface with the adhesive peptide, there were many more cells which adhered on that particular surface at the same time interval, 72 hours after the inoculation of the surface with the cells. All experiments were conducted with the same number of cells at the starting point. Still, a great section of the surface area of this substrate is bare, not covered by endothelial cells, under these chosen conditions for the experiment. For this reason, another experiment was conducted, namely that of stimulating the cells with the appropriate growth factor, basic fibrillous growth factor. On this new frame, you see a micrograph of cells adhering on the surface of the substrate 72 hours following the exposure in the presence of fibrous growth factor. The surface of the substrate is unmodified. Let us combine now the two things which we know promote the adhesive interaction of cells and proliferation. This is the result obtained when the substrate glass was modified with the adhesive peptide from laminin in the presence of basic fibrous growth factor. The cells not only proliferated, but they covered the surface area of the substrate. I agree that this particular coverage is not 100% intact coverage. There's still some small areas which do not have endothelial cells. I always argued that if it's not 100%, I'll say it's 95%. Given a little bit of a longer period of time, let's say more than 72 hours, I will expect that the proliferation of the endothelial cells will reach the point that this entire surface area of the substrate will be covered with an intact layer of endothelial cells. This is the desirable outcome of this particular experiment, that combine modification of the surface of a substrate and a chemical stimulus for the cells to bring about the function of cell proliferation. I will close this lecture with an acknowledgement to my former graduate students, Dr. KCD, Ms. Meredith Hassenbein, Dr. Lance Kam, and Dr. Dave Puleo, whose projects provided the results that I presented to you in this lecture. In addition, I would like to acknowledge the collaborative endeavors of my colleagues from Albany Medical College and from the New York State Health Labs in Albany, New York. This is the end of Lecture 16B with the title Protein and Cell Interactions with Materials, Applications.